Hello guys, welcome to my channel SwathiPharmaJnan. Today's topic is anti-tubercular agents. What is tuberculosis? Tuberculosis is a dreadful disease which is caused by the Mycobacterium tuberculi, and here we are seeing this topic is there in third form D medicinal chemistry and also in 6th semester of B pharmacy. So first we'll see the introduction of anti-tubercular agents. What is tuberculosis? What are the causes, symptoms, all the points we'll see now. The history about the tuberculosis is the disease name is tuberculosis. The causative organism is Mycobacterium tuberculosis. Only one drug is not used in the treatment of tuberculosis, but the organism is very resistant to any drug. Only one drug. The history of tuberculosis says that only one drug we cannot use in the treatment of tuberculosis. Why? Because the bacteria is very resistant to any of the drug which we will give. That's why they have started using the combination therapy with 2 or more drugs used to reduce or prevent the resistance of strains of tuberculosis. Modern era treatment started with streptomycin, which is an antibiotic. Isoniazid is totally a synthetic compound. So all these are the history regarding the tuberculosis. Next, choice of drug combination is based on how we can choose the drug combination for the combination therapy of tuberculosis is based on the location of the disease, where the location of the disease in the gastrointestinal system or in the pulmonary system or in the nervous system, neural TB. So based on the location of the disease, we will select the drug combination based on the susceptibility test results. If the patient can withstand with the drug combination or not. Based on that results, we will take the combination pattern of resistance in the locality. In that particular local part of the patient body, whether the resistance is strong or not. Based on that, we will select physical condition of the patient, age of the patient and sex of the patient, toxicity of the individual agents. If toxicity is more, then we have to toxicity is more for one drug, then we have to consider the other drug which is having less toxic. So like that we have to take the drugs, we have to choose the drug combination to avoid the drug interactions. So these are the various points for the drug combination choice. Next, classification of anti-tubercular agents. How we can define anti-tubercular agents? These are the drugs which are used in the treatment of tuberculosis or called anti-tubercular agents. Based on the therapy or usage, they are classified as first line agents, second line agents, anti-tubercular antibiotics. Under first line agents, pyroxenamide, ethambutol, isoniazid are the examples. Under second line agents, ethionamide, para-amino-salicylic acid and clofazimine are the examples. Under anti-tubercular antibiotics, rifampicin, cycloserine, capreomycin and viomycin are the examples. Now we'll see the structure one by one. Isoniazid. Here this isoniazid structure contains the basic nucleus pyridine. Pyridine contains CONH-NH2 is the side chain which is attached at 4th position. That is structure of isoniazid. Uses or it is a remarkably effective drug, one of the primary drug along with the rifampicin. We can use this isoniazid. Pyroxenamide we can combine with isoniazid. Ethambutol we can combine with isoniazid for the treatment of TB. If the patient has taken long-term usage of isoniazid, then the side effects will be it will cause peripheral neuritis, means inflammation of the peripheral nerves. GIT disturbances like constipation can occur, loss of appetite and hepatotoxicity can be observed in the patient. This isoniazid, a few important points about isoniazid is also called as isonicotinic acid hydrazine. Hydrazine formula is NH-NH2. This is NH2 is hydrazine. So that's why hydrazine, it is a synthetic antibacterial drug and it is having high bactericidal property, and it is very effective against both intracellular bacteria and extracellular bacteria. It is one of the primary drug used in the treatment of tuberculosis, and this will show the bacteriostatic property in non-replicating microorganisms. It will show bactericidal property in replicating microorganisms. The organism loses its acid fastness after the treatment with isoniazid, thereby involve or interfere in cell wall synthesis. In this microorganism, that is Mycobacterium tuberculosis, there is a mycolic acid which is present in the cell wall. When the drug will enter and it will lose this mycolic acid from the cell wall, then easily the drug can interfere with the cell wall synthesis. So that is how it will lose its acid fastness. Mechanism of action of isoniazid is it affects the electron transport and interferes with the conversion of NAD plus to NADH. This nicotinamide adenine dinucleotide will convert into nicotinamide adenine dinucleotide hydride. So this conversion is very much important for the electron transport chain. So from there citric acid cycle and various metabolic reactions and ATP will be generated, which is the energy for the further functioning of the body of bacteria. So here isoniazid will affect this electron transport by interfering with the conversion of this NAD plus to NADH. So INH will synthesize a fraudulent NAD plus. So it will incorporate in the place of nicotinamide and it will form a false NAD plus. Fraudulent NAD plus means false NAD plus. Fraudulent NAD plus means instead of nicotinic acid or niacin in the place of NAD plus, INH will be incorporated. That is how it will form false NAD plus. This fraudulent NAD plus is not utilized for normal oxidation and reduction processes in the cell because it is not a normal NAD plus. That's why it will not be involved in the oxidation and reduction processes in the cell. So isoniazid acts as an inhibitor of the conversion of C24 and C26 saturated fatty acids to C24 and C26 unsaturated fatty acids. Saturated fatty acids will be converted into unsaturated fatty acids by using one enzyme, that is desaturase. So this INH isoniazid will inhibit this desaturase enzyme so that conversion of saturated fatty acids to unsaturated fatty acids will be blocked by inhibiting desaturase enzyme. These unsaturated fatty acids which will be converted from saturated fatty acids are responsible for the or precursors for the synthesis of mycolic acid. Actually the mycolic acid is an important component in the formation of a bacterial cell wall. If mycolic acid is inhibited, the synthesis is inhibited, the bacterial cell wall cannot form and it will not show the protection, it will not give protection to the inner cytoplasm and nucleus. That is the mechanism of action of isoniazid. Here the flowchart form also you can see isoniazid which will enter into the cell wall, and this isoniazid will convert into isonicotinic acid, and it will incorporate in the formation of fraudulent NAD plus. So fraudulent NAD plus, which is a false NAD plus, will not show the process of oxidation and reduction process. No oxidation and reduction process will lead to no electron transport chain and then no energy for further functioning of the bacteria. Pyroxenamide. Pyrazine, the name indicates it is a pyrazine structure. Pyrazine means 6-membered ring containing 2 nitrogens which are at first and fourth position. Amide indicates amide group is present at third position. That is structure of pyroxenamide, which is a first line drug used in the treatment of tuberculosis, and it is having tuberculocidal activity which can kill the Mycobacterium tuberculosis. Next structure is ethambutol. Eth indicates ethyl group at the center. Arm indicates 2 amino groups. But indicates 4 carbon chain is present. C2H5, C and CH2, 4 carbon chain. Ol indicates butanol group. So 2 butanol groups are present and 2 amino groups are present. So this is butanol amino group. Butanol amino group both are attached with the center with ethyl group. That is a structure of ethambutal, which is very simple structure. By name of the name of the drug, you can remember the structure easily. This compound is only active against dividing mycobacterium, and it is bacteriostatic or bactericidal based on the conditions. Sometimes it will show the bacteriostatic property, sometimes it will show the bactericidal property, depending on the conditions of the mycobacterium tuberculosis. Its action is due to inhibition of incorporation of mycolic acid into the cell wall of the microorganisms. It has a selective toxicity towards mycobacterium. It will inhibit the incorporation of mycolic acid, which is an important component required for the formation of the cell wall of microorganisms. That is the mechanism of action of ethambutol. Thank you.